The cell cycle, the process in which a single cell divides and becomes two individual cells. Join us as we take a closer look. The cell cycle control system is made up of molecules that trigger and coordinate key events at certain checkpoints throughout the cell cycle. When you hear this sound, you'll know we've reached one of these checkpoints.
You're in interface. When you grow, I'll help you grow. Making proteins in 3 phases, G1, S, and G2 stages.
G1, or first gap, is where interphase begins. During this time, the cell grows and carries out protein synthesis and other cellular functions. The nucleus is completely intact, and individual chromosomes are not visible. The G1 checkpoint is here, also known as the restriction point. This point is very important to the cell because here it decides whether or not it will continue with cell division. If the cell is not prepared to continue with the processes of cell division, it will go into a non-dividing state called the G0 phase. The next phase is the S phase, or the synthesis phase. During this period, the cell replicates its DNA. All the chromosomes now have 2 chromatids. However, the chromosomes haven't condensed yet, so they're not distinctly visible. In the final stage of interphase, G2, the cell continues to carry out all normal functions and continues to grow. Near the end of G2, the cytoplasmic organelles replicate in preparation for the cell to divide during the upcoming process called mitosis. The G2 checkpoint is here. At this point, the cell decides whether or not it can move on to mitosis. It does this by seeing if it has enough MPF molecules, which are cyclin CDK complexes.
Just check out how cells split. They split by mitosis. Mitosis, what every cell knows is when it comes time to divide. Mitosis will be working inside.
Prophase. After the nucleoli disappears, this phase includes the coiling of chromatin fibers in duplicated chromosomes, appearing as 2 identical sister chromatids. They are joined at their centromeres and on their arms. The mitotic spindle begins to form, and centromeres move away from one another by the lengthening microtubules between them.
This is prometaphase, which takes place after prophase and before metaphase. In this phase, the nuclear envelope has almost completely dissolved and is made up of mostly fragments. Microtubules from the centrosomes, which are located at the top and bottom of the cell, attach to chromosomes, which are made up of 2 chromatids held together by a kinetochore. The centrosomes are more closely condensed in this stage as well. Microtubules jerk the chromosomes to get them in line for the next phase.
This here is metaphase, which is the longest stage of mitosis. You can clearly see the chromosomes line up on the metaphase plate, which is an imaginary plane that the chromosomes lie on in the middle of the cell. And the kinetochores of the sister chromatids are attached to the kinetochore microtubules coming from the opposite ends of the cell.
The M checkpoint is here. At this checkpoint, the cell receives the go-ahead signal once it has been approved that chromosomes are lined up along the meiotic plate and that the spindle fibers are properly attached at the centromeres.
Next is anaphase. This phase begins suddenly and lasts only a few minutes. During this phase, sister chromatids begin to part, each becoming a separate chromosome. Then the chromosomes begin moving towards the opposite ends of the cell, making the cell longer. By the end of this cycle, both sides of the cell carry the same chromosomes, and telophase begins.
Telophase. Microtubules push apart to elongate cells to its maximum size. Cytokinesis occurs to separate cytoplasm and its contents. Nuclear envelopes from the endomembrane system form around chromosomes. chromatin fiber proteins relax and DNA becomes less tightly coiled. 2 genetically identical cells result.
The process of mitosis is complete. However, errors and mutations can occur during these stages, and that's when trouble can arise.
Uh-oh, these cells are dividing at an extremely rapid rate. They aren't paying any attention to the cell cycle control system. These cells must be cancer cells. If they continue to multiply this quickly, they'll soon form a malignant tumor, which could potentially spread to other parts of the body through metastasis, and if gone undetected, will kill the organism. That would be bad.
Get to know the cell cycle. Interphase consists of 3 different parts. G1 is G2, that's how the cycle starts. The cell grows bigger in G1. It's time for a break. Oh wait, it's not done in the S phase. The DNA must replicate, and during G2 it's checked for vital mistakes. Mitosis, it's a cell division. This happens under chemical supervision. In prophase, the chromosomes have to condense, and metaphase this begins to make sense, cause the double-strand chromosomes have to line up. Yeah kids, that's what's up. And the next part comes the anaphase, where the chromatides and they separate. And telophase, man, it's so dope, cause it forms a new nuclear envelope. And last but not least, cytogenesis. I hope you understand, just listen to this, cause this is the part where the cytoplasm, man, it finally splits, huh? Yeah. Cell cycle rap.